Hello students, welcome to this session. Let's study infection. Definition of infection: it is the process in which pathogenic organism enters, multiplies and invade normal anatomical barrier of host, resulting in disease. An infectious disease, when clinical manifestations appear, it is called infectious disease. Now let's see types of infections. So primary infection is the initial infection. Reinfection is subsequent infection by same organism. Secondary infection, subsequent infection by new organism. Focal infection, it is localized infection. It can be superficial or deep. Superinfection, the second infection in already existing infection. Cross infection, new infection acquired from other host. Nosocomial infection, it is hospital acquired infection. Heterogenic infection, it is physician or medical paramedical staff induced infection. Depending on source, the types of infections are endogenous infection and exogenous infection. So endogenous infection, it is from within body of host. Example, normal flora. So two types of normal flora: transient flora and resident flora. So transient flora is present at some point of time, not always, and resident flora, they are present constantly. It prevents permanent colonization of body by other microorganisms, that are pathogenic microorganisms. The exogenous infection from outside body of host, so that is environment, so air, water, food, hospital equipments like thermometers, stethoscope, blood pressure apparatus, bedpan, urinal, trolley, etc. Then healthcare workers, other patients and carriers. Types of infections according to the clinical manifestations. So they can be asymptomatic and symptomatic infection. Asymptomatic when no symptoms are seen, and symptomatic when symptoms are seen. So it can be acute infection when the symptoms are seen for a short period, and chronic infection when symptoms are seen for long period, that is more than 6 months of duration. Latent infection when infection is in hidden form. Types of infectious diseases depending on the epidemiological pattern. So it can be endemic, hyperendemic, epidemic and pandemic. So endemic infection when it is persistent in particular area at lower rate. Hyperendemic, the infection occurs at much higher rate than the usual in particular geographic area. Epidemic when infection occurs at higher rate in particular area. Pandemic when infection spreads to large areas of the world. Example, COVID-19. Healthcare associated infection, that is hospital acquired infection, is defined as new infection occurred in a healthcare facility by a patient after 48 hours of the admission, which was neither present nor incubating at the time of admission. Community acquired infection or associated infections refers to the infections which developed in the community or within 48 hours of the admission to healthcare facility. Some infections can be localized. They are present in local area. They can be superficial or deep, and they can be generalized, like bacteremia, presence of bacteria in blood; septicemia, presence and multiplication of bacteria in blood; pyemia, pyogenic bacteria are circulating in blood, characterized by development of abscess in the various organs. Source of infection can be endogenous, that is normal flora. It can be transient and resident flora. Exogenous can be environment or hospital equipments or healthcare workers or other patients and carriers. Sources can be animals, so they are called zoonotic diseases if the infection is from animals. So bacterial infection like plague, viral infection like rabies because of dog, then protozoal infection, toxoplasmosis because of cat, helminthic infection, hydatid cyst because of dog. So these are animal acquired infections. Then insects, they are called arthropod borne diseases. So they are vectors, can be mechanical vectors like flies, they cause dysentery, typhoid. Biological vector, they cause Aedes mosquito cause yellow fever. So mechanical vector when there are no multiplication or the fertilization. Then biological vector when some sexual cycle present in that vector is called biological vector. Then other source can be soil and water. So bacterial spores can be tetanus. Then parasites like microorganisms causing vibrio, hepatitis, viruses. Then food, it can be external contamination, food poisoning or staphylococcal poisoning. then pre-existing infection, meat, animal products. Carrier, a person who harbors infectious agent in absence of clinical symptoms but transmits infection via contact or secretions. So the types are healthy carrier without suffering from disease, convalescent carrier who recovered from disease and continue to harbor and shed pathogen from the body, temporary carrier, they shed organisms for less than 6 months period, chronic carrier, they shed organisms for longer periods, contact carrier, acquire pathogen from patient, paradoxical carrier, acquire pathogen from another carrier. Methods of transmission: contact, like sexually transmitted diseases like syphilis, gonorrhea. Indirect contact can be fomites, inhalation, droplet nuclei, ingestion, waterborne like cholera, foodborne, food poisoning, handborne, dysentery, inoculation, so tetanus, rabies, syringes, needles causes HIV, hepatitis B, hepatitis C because of the blood transfusion. Then congenital, it can be vertical transmission, syphilis, rubella, or iatrogenic can be because of the lumbar puncture, catheterization, transfusion, dialysis, or organ transplantation. Factors responsible for microbial pathogenicity: so they are adhesion, so because of the fimbria they are adhesion to the host cells. Invasiveness, so it causes spreading, it can be localized or generalized. Toxigenicity, so it is because of the endotoxin or exotoxin, this we will see in detail later slide. The plasmid can be R factor. Bacteriophage, so beta or tox plus gene, bacteriophage in diphtheria. Communicability, spread from one to another individual. Other bacterial factors like thrombin-like enzymes seen in staphylococci, fibrinolysins, hyaluronidase, leukocidins, hemolysins. Bacterial appendages like capsule or bacterial VI antigen in Salmonella, K antigen in Klebsiella. Biofilm is a group of bacterial cells which stick to each other on a surface. Example, prosthetic valve. Infecting dose is minimum inoculum size that is capable of initiating infection. Route of infection, any route or specific route required. So these are the factors causing microbial pathogenicity. Now we will see endotoxin and exotoxin differences. So endotoxin, lipopolysaccharide in nature, exotoxin, proteins. The source, endotoxin form part of cell wall of the gram-negative bacilli, and exotoxin is secreted by gram-positive and gram-negative bacteria. The endotoxin released after cell lysis, exotoxin actively secreted by bacteria. Endotoxin heat stable, exotoxin heat labile. Endotoxin no enzymatic action, exotoxin action often enzymatic. Endotoxin non-specific, no specific tissue affinity, active very large doses and weakly antigenic, ineffective and not used for vaccine. Exotoxin specific action on particular tissue, specific tissue affinity, active in minute doses, highly antigenic, effective by neutralization antibodies, and toxoid forms are used as vaccine. Example, tetanus toxoid, diphtheria toxoid. These are my references. I think you can study from these references and I hope you find this session useful. Thank you.